All right, hi everybody. So, let's try to think what I want to talk about. Let's get this going. I'm just opening up Blackboard so I can show you where things are. I've got some of those wheat bags. My plan for the afternoon is just to either lay in a hammock or lie on the bed and read or snooze or something. But I'll get some of those wheat bags out and heat them up. But good idea though, thank you. Right, so essentially what we're going to be doing for this lab and next lab, right? So this lab can be kind of like a general introduction overview, I guess. And then if you work through it, I need to change the deadline. I'm going to do it for next Wednesday. Actually, let's do that now because I've got memory of a fish right now, to be honest. Give me just a second. All right, so just what I was doing just there is changing the due date of this lab, so the Eugene lab. I'll type that out as well. And so that's really just because there's an exam on next Tuesday, so I don't want you to have to choose between the two of those, and there's not really any need for you to have to do so. So it's the last module, module 6, correct? Yeah, so essentially what the idea is that you would have done this sequencing, and right, we've provided a whole bunch of sequences. Actually, let's just share my screen, then I can bounce around. Watch it. Different things. So the yellow sequences are here. Get out of there. Just here, it's a zip file. So download that to your computer or device. I would recommend making a separate folder, which you'll need to do for the Eugene assignment. And put it there, unzip it there, right? So this is a mix of sequences from wild type flies, so brown-bodied flies, and yellow flies. There's a mix of normal and mutant flies. There's also a nice little PowerPoint here if you want to gen up on Sanger sequencing. Wouldn't necessarily hurt. And basically what you'll be doing is walking through, oh, get out of there. This handout. So essentially what we're doing is kind of leading you quite gently, I think, through all of the different steps that you would take once you got your sequence data back from the sequencing center for whatever it is that you're sequencing, flies in this case. And really, this is, I think it's rather well done personally. This is breaking down all of those steps into their finest granularity, right? So, you know, where do you start looking for genetic information? Well, it's a fly, so you go to fly base, right? If it was a worm, you go to worm base. I don't know if they have mouse base. I don't think they do, but anyway. And that contains a whole bunch of info about that gene, right? That's been annotated and curated and added to. And typically, you know, you'll find papers that talk about that particular gene. You'll find sequences, phenotypes, all kinds of things, right? It's a very valuable resource. And then the first kind of part of this handout is really just going through these different steps, right? You know, you're going to be finding a gene, then looking at some of that gene information, right? Because there's lots of information on that page. And then answering some questions on Blackboard. So the lab module 6 questions, I'd actually suggest, you know, you start it and you just kind of go back and forth between the handout, the questions, and essentially what you're doing. And there's also, let's see. And it loads up. There are also 5 really nice videos that will also help walk you through this, right? So essentially what these videos are really in-depth step by step, I'm speaking right today, walkthroughs of really what you're being asked to do in the module. So if there's anything that you don't get, because some of this stuff is really quite straightforward, other stuff much less so, right? So I'd suggest having all of these things open, right? You know, the video from Dr. Page, which describes what you're going to be doing, the handout, which is kind of leading you through the different steps, and then the questions that you'll be answering, right? And each one of these questions corresponds to a question in that Blackboard assignment. And then bounce back and forth between them, right? So if you're wondering about the, you know, how do I figure out what alleles there are, right? Well, you know, go to that part of the video, watch through and see what Dr. Page does, and he will show you exactly what you should be doing. So then you do that, you go back to the assignment question, read through what it's asking you, and then you just answer the question, right? So it's a very deliberate walkthrough of all of these different steps. And I think it's been broken down very nicely into like each discrete bit that you need to do, right? And the hope is that, you know, trying to do this for the first time on your own would be really difficult, right? You know, I don't think anyone really starts doing this kind of work without any kind of help. And so really this is the equivalent of someone, you know, standing over your shoulder going, oh, you know, let's try clicking on that bit or try that bit, and just taking you through it. So even though you're not doing this really on your own, right, because there's a lot of not so much hand holding but guidance, the hope is that the next time that you need to do this, you would have already kind of had that experience, and so you'll know, okay, I need to find out this gene. Well, I need the reference sequence. Where do I get that from? NCBI. Go to NCBI, you know, you type in your gene, or worm base or fly base if you work on a model organism. And then you get that information, and then you can remember, oh, I need in a FASTA file because that's what's going to be recognized by my bioinformatics software, right? And so essentially this is really giving you the experience without you having to figure out any of this yourself, right? Because a lot of it is stuff that you don't know yet, essentially, which is why we're doing it. If you've done this a few hundred times, it's pretty trivial. We don't even really think about it. But we understand that you're, that's my Zoom video. You're at the beginning of this process. And so this is really a very well thought out walkthrough, essentially. And you'll see, you know, all of these different things, right? We're essentially getting you to look at some of the information that you find there. Where's that? Don't know if I've, no, I don't actually have that open anymore. But if you click on any of these, they literally go through exactly what it is that you should be doing at that point to answer this particular question. Right, you know, so you're going to do some interpreting of the visuals of the sequence, like, you know, what does this look like? What can we infer from these images? How do I find this sequence data? You know, where do I do it? And then the final thing is, well, two things. I think it's goal 4 and 5. Yeah, that's right. Oh no, 3, 4, and 5. 3 things. So the last half of this, right, which is actually in many ways the hardest part, is using a program called Eugene. And so Eugene is an extraordinarily powerful, but not especially user-friendly bioinformatics software package. It's very, very powerful. It's the equal of any of that I've used. But it's open source and free, so not as much work has been put into making it user-friendly. And also these things tend to be written by people who already know what they're doing, and so some stuff is a little bit more obtuse than maybe it needs to be. But essentially, this is going to walk you through getting those yellow sequences out. You're going to tweak them a little bit because they're raw sequencing files. And when you look at one, you'll actually see that there's like some mess essentially at either end, right, that you need to clean up. So that's what you'll be doing through here. And then once you have that nice and tidy, you're going to use the file that you downloaded from NCBI, which is the reference sequence file. And then you're going to see how well your actual sequences, the ones that you got, map to that sequence. Like where on the sequence does it match, right? Which is what you're going to be taken through here. Again, don't expect to be able to do this all in one sitting. There's quite a lot of work, and it pays to kind of take a steady approach to it, right? If you try just rushing through, it'll be fairly easy to make mistakes because essentially every single thing builds on what has come before it. Right, so if you skip ahead or you get something wrong, then it's going to make the next step a little bit harder. And then once you've you know, prettied up a bunch of those sequence files, and I'd recommend, if you can, do at least half, right, to make sure you have both wild type and mutant sequences represented. And once you've tied them up and made them kind of nice and neat and tidy, now you want to line them up, right? And so this is, if you remember back to when we talked about consensus sequences, particularly with regards to promoters like the Tata box and the like in prokaryotes. Essentially, what you're going to be doing is lining up a whole bunch of sequences, just as they did for figuring out that information. And you're going to see, particularly at one single base, which base is present there, right? And when you line up a whole bunch of them together, it's really, really obvious which one, because you should have the whole stretch of the sequence, apart from maybe some fluff at either end, should be identical between your mutant and your wild type sequences and your reference sequence. But there should be one change, and that change should be the same in all of the mutant sequences compared to the wild type. And so you'll be walked through that process. And then when you do, you'll have the final 4 questions to answer from 14 to 18. So remember, again, make the most of these videos. They're very good. They're very thorough. And they will take you through everything that you need to know to do these labs. Next Tuesday, I'll make sure I have Eugene open. Yeah, no, I don't have it open. Just I was playing around with it the other day. And some sequences up and ready to look at. So if you need any help doing the Eugene part, which I would say is probably the harder part of the assignment, then I'll be able to walk you through it in person. Well, you know, as in person as we can get. And then you need to finish all of those questions by Wednesday night at midnight. And so these essentially this is two labs, right? So this assignment is worth 100 points. And it's worth doing well, obviously, because you know that will help boost your lab grade, and which in turn will help boost your course grade. And that's pretty much it from me. So remember, we've got these videos. Get out of it. Move. Sorry, waiting for this bloody bar to go up. Here we go. We've obviously got this handout, which you will follow, you know, to the letter, pretty much. Your sequences are here. The sequences that we got back from the sequencing center. And here's your assignment that you'll be doing for next Wednesday. And that's pretty much all I have to offer. I kind of said it was going to be quick, but also I'm kind of like pie-eyed right now. So does anybody have any questions? Yeah. Yeah, I'm looking at my bed right now. It's looking very, very appealing, to be honest. I'm just going to lie down, close the curtains, maybe read a book. Cool. All right. Well, thanks very much, everybody. Sorry I haven't been quite on top of my shit today, but hopefully extenuating circumstances and all that. If you have any questions, as always, just shoot me an email. I'll get back to you as soon as I'm able to. And we also have the opportunity to go through this again on Tuesday too. And thank you for all your wishes. They're much appreciated. All right. Sayonara. Have a great weekend and see you next Tuesday. Oh, also, anybody wants to talk about any of the material before class on Tuesday for the exam, just send me an email. I can arrange individual Zoom meetings. They're really not much trouble at all. Cool. All right. I love you and leave you, as they say. All right. See you all. Well, I thought I ended it.